Hello everyone, welcome to BIO110 Ambassador Program. I'm Yvonne, and today I'm going to talk about how to teach bacteria new skills. So as you can see, this is a bacterium. And this is a plasmid. A plasmid is a small circular DNA molecule. It contains ampicillin resistance gene and part of the lexin gene. So let's look at it closer. First, look at the lexin gene. So if it is active, then the egg scale would be turned into blue. And let's look at the ampicillin resistant gene. It will help to survive an ampicillin. Now let's look at the transformation. So what is transformation? It is basically the process that bacteria taking foreign DNA. So after this process, the bacteria will gain new traits such as antibiotic resistance. But how, indeed, does bacteria take in this foreign DNA? Let's look at the experiment we had in our lab. So what we actually need to do is turn the bacteria into a competent one so that it would have the ability to take in foreign DNA much more easier. And step 2, we would add plasmid DNA. So we added the BIO110 plasmid to the competent cells. By contrast, the control group did not receive plasmid DNA. And we use heat shock at 42 degrees for 45 seconds. This step helps DNA enter the cells. After that, the cells were placed on ice to stabilize the DNA. Then is the recovery. So the cells were placed in LB broths and allowed to recover. This helps them express the new genes. And it will be at 37 degrees for 10 minutes. The fifth step is played on different media. So there are 4 different plates, which are LB, AMP, LB plus BIO110, and LB AMP plus BIO110. The upper 2 plates are the control group, as we did not add BIO110 plasmid. So they do not have the ability to survive ampicillin or turn egg scale into blue. And the LB plus group, as it does not have AMP, so the lexin gene would not be activated. As a result, only the LB AMP plus group would have blue spots on it. So as we can see the blue, this means bacteria took in the plasmid and they survived the ampicillin and the lexin gene was active. So in conclusion, if we see the blue spots, the transformation is successful. So in our lab, we successfully give the bacteria a new ability, that is to survive ampicillin. Our lab is a simplified module. It shows how scientists engineer bacteria, and we can use that to produce useful products. So let's see how it works in real life. Scientists first identify a useful gene, for example, a human protein. Then they insert this gene into a plasmid. the plasmid is introduced into the bacteria using transformation. The bacteria then express the gene and produce the desired protein. One classic example is insulin production. Scientists insert the human insulin gene into bacteria. After transformation, bacteria produce insulin protein. This insulin is then purified and used to treat diabetes. Before this technology, insulin was only extracted from animals. Now it is produced more safely and efficiently. In addition to insulin production, this method is widely used in many areas of medicine. The first example is antibiotics. First, bacteria can be used in the production of antibiotics. Scientists can modify bacteria to increase production efficiently and help us to produce on a large scale. And the second is vaccines. Scientists can insert genes from the virus into bacteria. The bacteria produce viral proteins, which are used to make vaccines. These vaccines help the immune system recognize and fight infections. And the third one is protein-based drugs. These can be enzymes or antibodies. They are used to treat different diseases. So thank you for watching this video. I also like to express my sincere thanks to my family for always supporting me, and to DK Youth for creating such a great learning environment, and to my instructor, Professor Lee, for his patience and guidance, and to my classmates for making this journey enjoyable. Thank you for listening again.